Hello, in this lecture we shall talk about C4 pathway. Photosynthesis, as you all know, is the conversion of solar energy into chemical energy. Terrestrial plants have been conveniently classified as C3, C4, and CAM plants based on the differences in the photosynthetic carbon metabolism. C4 plants are called C4 because the first stable product formed during photosynthesis is a C4 carbon compound, and hence it is called as C4 plant. This pathway is also called as Hatch and Slag pathway because for the first time Hatch and Slag found the C4 products in the sugarcane plants in 1966. This pathway is commonly seen in grasses, maize, sorghum. The first stable compound formed are the dicarboxylic acids, namely malic acid and aspartic acid. Unlike in C3 plants where phosphoglyceric acid is the first stable compound. C4 plants use the C4 carbon fixation pathway to increase their photosynthetic efficiency by reducing or suppressing photorespiration, which mainly occurs under low atmospheric carbon dioxide concentration, high light, high temperature, drought, and salinity. Which means under stressful conditions, a normal plant is made to behave like a C4 plant. C4 plant fixes more carbon dioxide as compared to C3 plants because of a special anatomy called as Crank's anatomy. There are roughly 8,100 known C4 plant species. Although only 3% of flowering plant species use C4 carbon fixation, they account for 23% of global primary production. Coming to the structural features of C4 pathway. As mentioned, they have a special kind of leaf anatomy called as Crank's anatomy. These plants do not show the differentiation of mesophyll cells into either palisade or spongy parenchyma. The most important feature of these plants is the presence of the vascular bundle in the leaf, which is surrounded by distinct parenchyma cells arranged radially in the form of a wreath, and hence it is called as Crank's anatomy. C4 plants are known to possess dimorphic chloroplasts. The chloroplasts in cells of the bundle sheath are larger and usually lack grana, while the chloroplasts in the mesophyll cells are smaller and always contain grana. Another important feature of Crank's leaves is the close packing of the mesophyll cells with very less intercellular spaces. Thus, this helps in easy diffusion of carbon dioxide to the carboxylation sites. Mechanism of C4 cycle. The four important steps involved in C4 cycle are fixation, transport, decarboxylation, transport. So here in the picture you can see the occurrence of the C4 pathway, which takes place in the mesophyll cells as well as in the bundle sheath cells. In the first step, that is fixation, carbon dioxide is fixed by phosphoenol pyruvate in presence of water and enzyme PEP carboxylase to give rise to oxaloacetic acid. Oxaloacetic acid is the first compound formed, which is unstable and hence it readily gets converted into either malate or aspartate. Malate is produced by the activity of malic dehydrogenase in the presence of NADPH plus H plus ions. In the second step, that is transport, the C4 acids which are produced in the mesophyll cells are now transported into the bundle sheath cells. So from chloroplast of mesophyll cells, the malic acid is transferred to the chloroplast of the bundle sheath cells. In the third step, that is decarboxylation, the malic acid, which is now in the bundle sheath cells, will undergo decarboxylation to release carbon dioxide and give rise to pyruvic acid in the presence of NADP plus, in presence of the enzyme malic dehydrogenase. Carbon dioxide released by the decarboxylation of malate is fixed in the bundle sheath cells by the typical C3 pathway, while NADPH travels back to mesophyll cells where it utilizes the light generated ATP to produce PEP again. In the fourth step, that is transport, the C3 acid produced in the bundle sheath is now transported back to the mesophyll cells to produce phosphoenol pyruvate, which now readily accepts another molecule of carbon dioxide. Thus, in C4 plants, we see two carboxylations taking place. The first carbon dioxide being fixed in the bundle sheath cells, and the second carbon dioxide fixation taking place in the mesophyll cells. Thus, this pathway is also called as dicarboxylation pathway. Significance of C4 cycle. 1. It increases the photosynthetic yield 2 to 3 times more than the C3 plants. C4 plants perform a high rate of photosynthesis even when the stomata are almost closed. C4 cycle increases the adaptability of C4 plant to high temperature and high intensities. They can grow very well in saline soil because of the C4 organic acid. In this particular slide, you can take a look at the differences between C3 and C4 pathway.